So in this exercise, we'll be covering tools of the laboratory. So within the micro lab, we have what we call the 5 I's of microbiology, which are inoculation, incubation, isolation, inspection, and identification. So the first I, inoculation, is technically the introduction of an inoculum into media to culture microbes. But let's break that down. So during an inoculation, you're going to introduce an inoculum into media. So what's an inoculum? An inoculum is just a small sample of microorganisms. It could be a small sample of bacteria or viruses or fungi, whatever it be. And since microorganisms are ubiquitous and found everywhere, a sample or an inoculum could really come from any possible place imaginable. Definitely in a healthcare setting, they're from these kind of standard places, but in the context of something like our lab, they could even come from surfaces such as inanimate objects, or the skin, or potentially even things like food and water. So inoculation is the introduction of an inoculum, or a small sample of microorganisms, into media. So what's media? Media is essentially food for microorganisms. So the media contains the nutrients that are required for bacterial growth. So they can feed off of those nutrients in order to gain energy to continue to grow and divide. So we know that inoculation is the introduction of an inoculum into media or food to culture the microbes. So what does it mean to culture the microbes? That essentially means to grow the microorganisms. Anytime you're asked to culture something, it is essentially you being told that you need to grow something. So once we've placed the microorganisms into the media or into the food, and they've grown, at that point, that growth is known as a culture. And there are different types of cultures. There's pure cultures, mixed cultures, and contaminated cultures. So a pure culture is going to contain only one species of bacteria. So if we look at this plate here, we can see that the growth is consistently the same color. These colonies all look the same, which is a good indication that this is a pure culture and only contains one single species of bacteria. Over here, we have what we could call a mixed culture. Mixed cultures contain 2 or more species of bacteria. If we look at this plate, we can see that there are different colors of colonies, different shapes of colonies, colonies with different outer edges or margins, and this is a good indication that this plate has different species of bacteria growing in it, which would make it a mixed culture. Then over here, we have what we call a contaminated culture. The main thing with contaminated cultures is they contain unwanted growth of some kind. So likely this individual is trying to grow these bacteria here, and if we look, we can kind of see what appears to be all this most like fuzzy growth, which is typically consistent with fungi. It looks like some type of airborne contaminants got in and continue to grow. So contaminated cultures just contain any type of unwanted growth. The media or the nutrients that are used can come in different types of preparations. So sometimes we use test tubes to store a liquid media. So it's just a glass test tube with liquid water-based contents, and the nutrients will be in the liquid form, and then the bacteria will just grow in the liquid. More often than not, though, we grow bacteria in petri dishes with a solid media. So the media used in this for the nutrients would be in the solid form. Kind of imagine just like a jello consistency type material that would be in a plastic dish, and then the bacteria would grow on top of that. We often do need devices to transfer microorganisms from one place to the next. For example, if you're collecting a sample from a surface, you would need to transfer that into a petri dish or a test tube, or maybe you're transferring bacteria from a test tube to a plate. In those instances, you would need some type of transfer device. So loops and needles are very commonly used. These are reusable transfer devices that you would need to sterilize before and after. An example of a loop can be seen here. This is an inoculating loop that's being used to transfer bacteria. Needles are very similar to loops. They just don't have this loop at the end, and they're just a straight point. And then we also use sterile cotton swabs. They come packaged in a sterile way, so they don't require any type of sterilization before and after like the loops and needles do, where you typically have to sterilize with heat. Regardless of what type of dishes you're using, or test tubes or transfer devices, you always want to make sure everything's done in a sterile way, because we want to avoid contamination as much as possible. And that includes contaminating our cultures and potentially contaminating ourselves. So we know with the media or the nutrients, it can come in test tubes or it can come in petri dishes. Typically when it's in a test tube, you're going to see it in a liquid form, which is what we see here. This is just a liquid broth, so it contains the nutrients in a liquid form that's in a test tube. Typically when bacteria are not present, this liquid will be quite clear. However, once we've grown bacteria in the broth will get very cloudy because of bacterial growth. And in micro, we don't really say cloudy. We say the solution has turbidity or is turbid. But anytime a broth is turbid, that is a good indication that bacterial growth is present. Sometimes we will have like a semi-solid media in these test tubes, which is somewhere between a solid and a liquid. Imagine a jello that hasn't completely set. We will often use this type of test to inoculate bacteria into and see if they were able to move throughout the semi-solid media, which provides us information regarding motility and the presence of motor appendages like flagella. And then most often we have solid media, which is what we're seeing down here. So this is just a plastic petri dish, and inside of the plastic petri dish there is just a solid media, so like jello that has completely set, but just a solid media that has all the nutrients for the bacteria that they'll feed off of, and then they will actually grow on top of the solid layer as we're seeing here. What actually makes the media solid in a petri dish is this substance called agar, which is derived from algae. Agar has absolutely no nutritional value. The only thing it does is turn a liquid into a solid. So really the only difference between this nutrient broth here and this nutrient agar plate here is the presence of agar. They both have the same types of nutrients. It's just that this broth does not have agar, and that's why it's in the liquid form. And then this plate does have agar, and that's why it's in the solid form. So oftentimes we'll use nutrient agar to grow bacteria. It kind of just is your generic all-purpose agar that's used to encourage the growth of all bacteria. However, if you have specific intentions with the bacteria that you're working with, you may need to use a more specific type of plate or media. So there are different media that we can look at based on purpose. There's selective media, differential media, and enrichment media. So with selective media, there is some type of inhibition process. With selective media, these are plates that are going to encourage the growth of some bacteria while inhibiting the growth of others. So it's kind of in the name. In a selective media, the plate is going to select for the growth of specific types of bacteria, and then others would be inhibited. So the best example of this is a plate known as an MSA plate, or mannitol salt agar. This is a plate that has a high salt concentration and will typically only allow the growth of Staphylococcus bacteria. So a lot of different types of bacteria. Most bacteria are not going to grow well in a mannitol salt agar, and that's because of the high salt concentration. Most bacteria, when you expose them to salt, they will shrivel and die. The exception to that rule is, or are, any type of Staphylococcus bacteria. They love salt because they're well adapted to surviving on the skin where there's a high salt concentration from sweat. So they're going to grow very well on this plate. So if we were looking at a mannitol salt agar plate, which we see right here, if we put two different types of bacteria in this plate, because of the inhibitory process, only one would grow. So in this plate, we've put Staphylococcus aureus over here and Serratia marcescens over here. We put two in, only one has actually grown though. So the Staphylococcus aureus was able to grow because it enjoys a high salt concentration. The Serratia marcescens though was not able to grow, likely because it was dehydrated due to the high salt concentration and shriveled up and died. But the main thing to understand is that in selective plates, you're going to be inhibiting the growth of some bacteria while encouraging the growth of others. So there's an inhibitory process. With differential media, there's no inhibitory process. If you put any type of bacteria in a differential plate, it's going to grow. However, the bacteria will grow different colors based on how they interact with the contents of the media. Typically, when they interact with the contents of the media, there's some type of observable change, usually a color change that's due to changes in pH. But the main thing is everything's going to grow, but it will grow different colors based on interaction with the media, which usually tells us something about the bacteria. So the best example of this is a blood agar plate, which is what we're seeing here. We've put 3 different types of bacteria in the blood agar, and all 3 have grown. There's no inhibitory process. But if you see, they've grown different colors. This one's like a pale green. This one kind of has like a clear halo around where the growth occurred, and then this one just growed kind of white on top of the blood agar. This does tell us something about each of these bacteria. Specifically, it tells us if these bacteria are able to destroy red blood cells or not. But the general premise with this right now is that everything has grown, but things have grown different colors based on interaction with the media, which again does tell us something specific about the bacteria. Now there are enrichment media. It's not something that we particularly use in this course because enrichment media is media that has been infused with specific nutrients used to cater the growth of very specific bacteria. In our course, we use very generic bacteria that don't require any type of specific nutrients. They typically just grow just fine in something like a nutrient agar. But if you would go out and identify an unknown bacteria, and that bacteria requires a very specific sugar in order to grow, you may take a nutrient agar and infuse it with that special sugar. At that point, the nutrient agar becomes an enrichment media since it has been infused with that specific nutrient that is meant to cater the growth of your specific unknown bacterium. So enrichment media is used to cater the growth of very specific bacteria. Now, it's not always straightforward. Not all plates are just selective or just inferential. There are often many plates, and even ones we use in our class, that are both selective and differential all at the same time. And what that means is they only allow certain types of bacteria to grow. That's a selective property. But the ones that grow different colors based on how they interact with the media. So the best example of this is something called an eosin methylene blue plate or an EMB plate. This plate will only allow gram-negative bacteria to grow. Gram-positive bacteria are inhibited, so only gram-negative bacteria can grow. But there are a lot of different types of gram-negative bacteria. Gram-negative bacteria will grow on here though, but they will grow different colors based on how they interact with the contents of the media. So here, these are all, well, most of these are gram-negative bacteria. They've grown. Down here, this one's gram-positive, so it wasn't able to grow. But these ones are all gram-negative bacteria, so they have grown. But growth can occur different colors based on interaction with the media. So we have some metallic green growth, and then we have some kind of light pink growth here. And that typically tells us something about how they interact with the media. In this one specifically, it tells us about lactose fermentation. But just keep in mind, plates can be both selective and differential, and they can only allow some bacteria to grow, but those that grow will grow different colors.